Hello, myself Ruchi. Today I am discussing about two topics. First is mechanism of photosynthesis and second is vision. Uh, Mechanism of photosynthesis, the general scheme of the coupling of various processes taking place in thylakoid membrane during photosynthesis. This scheme was proposed by Witt in 1971. The event number is followed as a 10 type. First is the migration and dissipation of energy. The energy is stored and the dissipate in the excited carotenoid car and also as a result as fluorescence. And second is the rate of reaction is photosynthesis first and second. Third is the generation of an electric field as the result of directed electron transport at right angle to the membrane, which is governed by the anisotropic orientation of the molecule obtained in the membrane. Fourth is electron transport and then influx of protein into the membrane in accordance with the Mitchell's theory. Two electrons on the outer side of the membrane are replaced by two alcohol ions and two holes positive on the inner side by two proton. The possible mechanism involves proto electrolyte reaction coupled to electrons transport from hydrogen bond to the NADPH. Fifth is the generation of pH. The bond alcohol plus hydrogen can be replaced by calcium and sodium. The the ions diffuse respectively into the external and internal phases, thus creating pH. Sixth is the field controlled influx of hydrogen and the decrease of pH. Seventh is the field controlled exflux of sodium. The diffusion controlled decrease in pH. Ninth is phosphorylation coupled to the discharge of electrolytic energies membrane is accordance with the Mitchell theory. 10th is the regulation of the inner wall pH. The potassium conductance of the membrane it is greater than hydrogen conductance. During the generation of hydrogen in the increase show down the electron flux which is turned retarded the generation of hydrogen. The increase is in exchanges the influx of hydrogen which occur due to the influx of sodium. The concentration is maintaining at a certain level in increase within increasing flashes frequently or light intensity. Processes 6 and 9 are also accelerated, thus the energy of light quanta charge the membrane. The right part of energy is used. Photosynthesis is a complex series of photo and electrochemical processes whose detailed mechanisms are still unknown in many respects. The Witt is so far hypothetically in quantitative estimation in the application of the Mitchell theory of photosynthesis is based with difficulties. In this theory, the free energy of proton must exceed the energy of phosphorylation. Migration of energy are reacted by dispersion of energy as reacted by the light reaction formation of field to electron transport and one revolution. Must not be less than experiment given a close estimated for one molecule of ATP to the synthesized by their required 4 proton on the other end from the reaction ATP. It's follow the two protons are consumed by the synthesis of one molecule of ATP. This energy is equal to 0.36 less than that the required for ATP synthesis. An inequality is not fulfilled. The energy of one proton is influenced. This description reveals itself more decently under the condition of pulse. It stimulated further investigation are needed for the Mitchell theory to be applied the phosphorylation in photosynthesis. Let us consider in conclusion the elementary process that control photosynthesis. Their classification has been proposed by Bossev in 1973. Following 4 classes of phenomena are to be considered. First is the absorption of light by chlorophyll and accessory pigment. Second is the migration of energy for light absorption molecule to the reaction center. Third is the primary separation of charges of different signs. Fourth is the stabilization of new energy carrier for coupling with the sequence chemical state. First group of phenomena has been theory studied and described above. Let us now turn to the second group. It has been stabilized that energy migration really exists and occur by the resonance mechanism. Other mechanisms of energy migration are also possible in principle. In the semiconductor mechanism, the excitation of the molecule by light transfers an electron into the conduction zone, after which a pair of opposite charges is displaced is an order molecule system in a molecule crystal. However, in chloroplast, strict order is characterized of only small assemblies of chlorophyll molecule. It is for the reason that the semiconductor mechanism is hardly probable at the same time molecule order in the chloroplast. Insufficient for the migration of molecule or order of chlorophyll is sufficient of the migration of the energy by other mechanism. There is possible a migration of energy triplet excited state or a resonant migration of the excitation and induction type. In the last two cases, a diploid interaction leads to the transfer of the energy of a singlet excited state to an unexcited molecule. In exciton migration, it occurs during time period comparable with the period of vibration. It takes 10 to the power 12 to 10 to the power 15 second to distance of up the 1.5 to 1.8 nanometer. This is the most rapid mechanism. The inductive type migration is preceded by the attainment of the thermal equilibrium of excited molecule with the medium, and the rate of migration strongly depend on temperature. A number of facts and in favor of rapid exciton mechanism. The primary physical step of photosynthesis begin in the absorption of a quantum and ending with the capture in the reaction center. Occur during the time not longer than about 100 nanometer. The time is the 1.9 to second order less than the characterized time for fluorescence of the dye molecule. It is the maximum time of diffusion controlled reaction. Such a rapid energy migration provide a high quantum yield to the primary photosynthesis process. The probability of a nature of the component of the electron transport chain has not yet been solved. Various scheme of the electron transport chain has been proposed, and important specific feature of the electron transport chain has been included, namely that the rate of electron transport is independent of the temperature at the low temperature. This has led to the natural assumption of the tunneling mechanism of the electron transport between neighboring carrier. The conception has been developed in the work of Blumenfeld and the Chernyshevsky. Electron transport occurs between the center fixed on in the thylakoid membrane. This center may be treated as sufficient to be deep electron traps. One may visualize such as a center of potential well and consider the level at that the electron being transferred occupied. Here one Here one deal with the problem of electron transport between two neighboring component of the chain. Example like between the principal level of potential wells separated by the barrier. In such a case, in the photosynthesis system, there occur electronic conformational interaction which can conventionally that by treated by resorting to the potential well model. After the electron transport has taken place, the acceptor molecule is found to be in a non-equilibrium conformation, which is slowly relaxation of equilibrium. This makes possible a balanced resonance and a tunnel effect. No strict quantitative theory has, however, been worked so far. My second topic is vision. Vision, apart from photosynthesis, another most important photobiological process in photoreception, which considered in receiving information about the factor of the outside world by means of light radiation. Let us consider the most perfect for a photoreceptions, the vision of vertebrates. The structure of the eye is showed the schematically. The eye is a self-regulating system. Its optical shortcomings are the considered extended compensation by regulatory mechanism that optimize the operation of the eye. Most important of these are focusing on an image of the retina and the regulation of the amount of light that falls on the retina. The focusing on spanning up of the image on the retina is accomplished through automatic changes of the radius of the lens curvature via accommodation. The shape of crystalline lens is controlled by a ring of muscles surrounding the lens. These are called the ciliary muscle. The contraction of relaxation of the ciliary muscles occur in the response of the diffusing of the image. The accommodation system is a scanning system since it's bring the image of distant or nearly object into sharp focus. To optimize the functioning of the eye, it is necessary that the amount of light that enters the via the lens of be regulated. What is actually regulated is the extent of opening of the optimal structure the lens. In bridge light, the lens of narrow. In a dim light, the lens is winded. This is affected by two antagonist muscles that form the structure of the iris diaphragm. The phenomenological interpretation of such a system is based on the general propagation of the theory of control and input signal that enter the system. Is different between from the required. The difference between the input signal and the setting is the error signal. The purpose of the regulation device consists in receiving the required output signal. The regulating system consists of a control and an object to be controlled. It contains a feedback circuit. Let us consider the structure at the retina. This is multi-layer structure. Show schematically an image is produced on a pigment epithelium. In outer layer 3 there is realized a synaptic coupling of photoreceptor cell to rods and cone. With a nerve horizontal cell 4, the other nerve cells bipolar cells and amacrine cells 6 are connected synaptically in layer 7 with the ganglionic cell 8, which serves as direct source of impulses that arrive at the axon of the optic nerve. The input signal is the optic images on the pigmented epithelium, and the output signal is the natural image coded by the impulse in the optic nerve. For light to reach the photoreceptor, it must pass through the layer of nerve cells. The photoreceptor cells are thus protected against harmful effects. The light sensitive receptor cells, the rod and cones are extended structure. The rods are long and narrow, and the thick cones are short and thick, made of many specialized compartments that form a sequence of parallel discs. The rod and cone of the frog are showed symmetrically in this disc contain photoreceptor molecule devices. The required substance are delivered via flagella to the disc for the basal body of the cell. Light is absorbed in the disc by the molecule system described in the next section. It has been found that the protein of the disc are continuously renewed. It is an stabilized effect. The cones are responsible for color vision, and the rods are sensitive to low level of illumination. As far back as 1756, Lomonovos wrote about the visual perceptions of three colors, three species of the either practical red, yellow, and blue. The origin of light in 1956. In the 1802, Young proposed a theory of color vision based on the assumption of the excitants of three types of light sensitive substance in the retina. The theory of color vision was further developed by Maxwell and Helmholtz. This theory has been supported by direct measurement of light absorption by individual cones. There are three types of cones characterized by different curves of absorption of photopigmentation. The curves for man are very similar to them. The receptor of invertebrates are arranged in a different manner. In many cases, they are sensitive in a wide spectral region.